Hello everyone, welcome back to our channel BioLab, and this is VAMSI. Yes, you might think that why I am using a new type of lecture videos. Since this is only for some time, as you have to understand sliding filament theory very much carefully, and even my tablet is also not having charge. It is not I did not even prepare the notes for it, so that is why I'm using this technique. So only for this lecture, or some, if you like this type of lectures, then I'll be continuing it. If not, we will be using the same the previous one only. Okay, today let's discuss about sliding filament theory. This theory tries to explain about muscle contraction. So let me explain you first of all. See, as the name of the theory, let's understand the name of the theory. What the name of the theory suggests: sliding filament. Here filaments are the actins and myosins. Myosin filaments. Actin filaments and myosin filaments are the sliding are sliding. Okay, sliding means passing one over the other, passing by, sliding. Yes, which pass by which? Does myosin filament pass by actin filaments, or actin filaments pass by myosin filaments? This theory tries to explain this one. Okay, now let's see the main key points on this theory. This theory was given by AF. So this is important. Write each and every point which I write on the paper. Proposed by AF Huxley and H.E. Huxley. AF Huxley and H.E. Huxley. These are the two scientists who proposed this sliding filament theory. Okay, who? AF Huxley and H.E. Huxley. Now let's understand the steps in muscle contraction. Before starting that, I have to explain you about two more proteins in actin. Okay, in actin filaments, I have to explain you about two more proteins which I haven't explained you yesterday, since there was no time. Now let me explain you. Without that, you will not get this sliding filament theory. Now let's move on to the two filaments in actin. Yes, we have discussed that actin is of two types: F actin, filamentous actin; G actin, globular actin. This F actin is made up of globular actin. Now, not only these actins, these F actin and G actin, there are two more proteins which are responsible for muscle contraction. They are troponin and tropomyosin. These are the two proteins which are responsible for this sliding or muscle contraction. Without these two, muscle contraction, you can say, yes, muscle contraction happens, but muscle relaxation does not happen. Okay, muscle contraction happens, but muscle relaxation does not happen. Yes, why we discuss in actin? These two are present on actin. They are present on actin. These two are present on actin. Actually, yesterday we have seen that actin, F actin, or actin, these two F actins are arranged helically, wounded to each other like this. Yes, yesterday I have shown you the diagram too. I am again doing it. They are helically round to each other. They are helically bound to each other. Yes. See, like this. The black one is one filament actin, the blue color one is one filament actin. There, where are these two present? Where are these two present on actin? Let me show you the presence of the troponin and tropomyosin. First, let me show you the presence of tropomyosin. This tropomyosin is present along the length of this actin. This tropomyosin is present along the length of these actin filaments. It is shown like this. See. Something what like this, you know, see, something what like this. This is tropomyosin. This is tropomyosin, this purple colored strand which is present along the length of actin. But whereas coming to troponin, this troponin is present as hope you cannot see this. No, this color isn't visible at all. Let's use brown. See, they are present like this on tropomyosin. They are present like this on tropomyosin. So these brown color ones are troponins, and this purple color one is tropomyosin. Sorry. This purple color one is tropomyosin, and this brown color ones are troponins, and purple color ones are tropomyosin. Hope you are getting it. Yes, this is how these two extra proteins which help in muscle relaxation are present on the actin filament itself. Now let us discuss some mechanism or some points related to sliding filament theory. This theory states that, let's write the statement here. I am giving you a simplified statement. It states that, this theory states that during muscle contraction, the actin filaments slide pass over the myosin filaments. Understand the meaning. Now take for example, I'm giving you an example here. Look at here. See, think this is actin. This blue color one is actin. This purple color one is myosin. We have seen yesterday this actin filament is present over the myosin. It is overlapping some part of myosin. Okay, let me remove this. Again I'll get it, please do not worry. Yes, see, this is actin filament, blue color one. This purple color one is a myosin filament. Yesterday we have seen this actin filament slightly overlaps the myosin filament. During muscle contraction, what happens is this head of myosin binds with the active binding sites at actin, and then like this, it happens like this, and during muscular contraction this actin slides over the myosin. The actin slides over the myosin. Now here you can observe that myosin length is getting reduced. See, before it was like this, it was this way. Let us mark that too. So before it was something like this till here. This is the before length. This is the before length. Let's say L, before length. But now, when this actin filament slides over the myosin filament, let's say till here it slided. Now this becomes the length. Let's say this L dash. I'm not teaching you maths or physics, just I'm explaining you the length has decreased. That length has been decreased of which one? Myosin filament, due to the sliding of actin filament. So how much length is decreased? From here till here, it is a decrease in length. It is a decrease in length. Okay, are you getting my point? Yes. While actin filament slides over the myosin filament, then the muscle contracts due to the decrease in myosin filament. And also while this actin is moving, yes, yesterday we have seen that actin is attached to two lines, yeah, Z lines, one on this side, one on this side. Let us draw the Z line also. Z line, in your language it's Z line. Okay, this is one Z line, this is other Z line. Here is one more actin. I am not showing it, that just I am showing you this. This is the myosin. Now see this. This actin is attached to the Z line. Actin is attached to the Z line. Now what happening is, let us place something also here in order to understand that. Okay, this cap, think this cap as Z line. Just for some time, think this black color cap as Z line. This black color cap is Z line. This blue color one is actin. This purple color one is myosin. Now, according to sliding filament theory, during muscular contraction, yes, I will be explaining you the mechanism how this myosin head binds with the active binding sites on actin. See, on myosin head, yesterday we have seen that there are active binding sites of actin. Yes, this actin binds. This actin binds when the actin. I will be explaining. Don't worry. This, think that it binds. That's it. For now, it binds. For now, this binds. Actin has binded. Now this is black color one is the Z line or Z line. Okay, this black color one is the Z line. Now what happens? Actin slides over the myosin. See, actin filament is sliding over the myosin filament. What is happening here? Did you notice something? Not only the actin moving, but it is also bringing the Z line towards itself or inwards. See, before the Z line was here, when actin was here, Z line was here. When actin was contracting, the Z line was also moving towards. So that happens on both the sides. Now this Z line length also decreases. The gap between them decreases, which means Z line, the unit which is present between two Z lines, we have seen yesterday, sarcomere, the length of sarcomere is contracting or it decreases. Therefore muscle is contracting. Muscle is only contracting due to the movement of actin, due to the sliding of actin. You have to remember that, due to the sliding of actin, or due to the sliding of thin filament over thick filament, remember, not light filament over dark filament, thin filament over thick filament. Thin actin filament is thin, we have seen yesterday. Thick myosin filament is thick, we have seen yesterday. The thin filament sliding over the other. When it is sliding over the other, it is also bringing the Z line along with it, so that it is contracting. Now the Z line's position is this one. See, length has been decreased. Length of the Z line is also decreased. This is one Z line, this is one Z line. This is Z dash, this is Z. Z dash is the new position when the actin slided. L dash is the new length of myosin when actin slided. Z was the initial position when actin wasn't in sliding position. Here also same. Here L was the initial length of myosin. Okay, on both sides the same thing happens, but I have shown you one side. Okay, got it? No confusion, no confusion. Now let's move on to how this sliding gets initiated. First of all, who is the one who initiates this sliding? Yes, for initiation of sliding, we require signal from the CNS, central nervous system, or our brain. Okay, or our brain. See, our brain, CNS, using somatic nervous system, sends signal to the neuromuscular junction. This is the muscle. This is the sarcolemma of the muscle. Sarcolemma, yeah, plasma membrane, sarcolemma, sarcoplasmic reticulum, endoplasmic reticulum is called as sarcoplasmic reticulum. Yes, we have seen yesterday, sarcolemma of the muscle. Now this is the neuron. This is a neuron. Now when this CNS sends information to the somatic nervous system, it sends message, then this message in the synapse, of this is called as synaptic cleft. We will be seeing in neural control and coordination chapter, which is the second chapter after this one we will be seeing that. So this is the synapse. Now this thing which is get here, that is called as nerve impulse. The nerve impulse is released as neurotransmitters. These are neurotransmitters. Generally a neurotransmitter in somatic nervous system is acetylcholine. Acetylcholine. Remember it. Acetylcholine is the neurotransmitter. It is released at the junction, neuromuscular junction, and neuromuscular junction is the junction between motor nerve and this sarcolemma, or the muscle. Okay, neuromuscular junction. Now when this acetylcholine is released, the T tubules which are present sides to the Z line. See this is the Z line. The T tubules are present like this. These are T tubules. They release calcium ions. Yes, the sarcoplasmic reticulum is a storehouse of calcium, Ca+2 ions, and these T tubules release them. When these T tubules release the what we call calcium ions, then these calcium ions are very much efficient in binding with the troponin and tropomyosin. Yes, these two, they are very efficient in binding with troponin and tropomyosin. Actually what happens is this troponin and tropomyosin mask the active binding sites of actin. See, if this is the actin, say this is actin, here troponin is present and tropomyosin is passed over it like this. They mask the active binding sites. In order to remove this mask, as myosin has to bind with the actin, myosin head has to bind with the actin, this calcium ions joins with this troponin and tropomyosin and removes the masking. Here now there is no masking. Now actin filament is free to join with the myosin filament. Now what happens is this myosin filament. So see this actin, this is myosin, this is head of myosin. Now this head of myosin binds with the actin and it pulls towards it. It pulls the actin towards it. It pulls the actin like this. See, let me tell you. I am giving a basic explanation about this. Let me tell you once wait. Let me tell you. It pulls the actin. See, how to explain you. Yeah, this is the actin, the previous explanation page. Let's go into that. I'll bring it again. This is actin, this is myosin. Think there is a head for myosin. Let us draw the head for myosin. This is the head for myosin. Think for some time. This is the head for myosin. Now here the actin will not, or the myosin head will not bind with the actin unless and until the masking is removed. Now wait. That is why I have selected this type of lecture for this sliding filament theory. That is very important. Now see, this chalk piece I am putting here. Now think, for suppose this chalk is the troponin. It is not allowing the myosin head to bind with it. It is masking. Now calcium ions enter. Ca plus ions. Before Ca plus ions, what happens? Neurotransmitter is released. With the neurotransmitter release in the neurotransmitter, Ca2 plus ions are released. Now with the entry of Ca2 plus ions, this, what we call this troponin, binds with the Ca2 plus. It got binded with the Ca2 plus. Now unmasking is done. The masking of troponin and tropomyosin is removed. This is the masking. Now it is binded. Now move aside. Now this head binds with actin. The head binding is completed. Now as this head is like this, see, if this is the head, this is the actin, the head is binding like this. In order to retain its position, or in order to pull this actin towards itself, it gets its head backwards. It pulls the actin, see, like this. It is pulling the actin towards it. Head is pulling the actin towards it. Now the head is pulling towards it, the actin towards it. Therefore the actin slides over the myosin. If actin slides over myosin, Z line also slides. If Z line or Z line contracts, sarcomere contracts. If sarcomere contracts, muscle contracts. If muscle contracts, there will be a movement in the muscle. Got it? No confusion? Yes. Now come. We have to see some terminology. Let's discuss some terms. First one is cross bridge. Now what is this cross bridge? Yes, when myosin head binds with the actin, this is called cross bridge formation. I am not writing any definition. Just remember, get into your brain. What is a cross bridge? A cross bridge is formed when the myosin head tries to bind with the actin active sites, in order to move the actin towards itself. Now after this cross bridge formation, this myosin pulls this actin towards itself. If this is myosin, it pulls actin in this direction. Actin slides. Now in order to give a power stroke means pulling. This is power stroke power stroke. This is called as power stroke. It is pulling towards itself. In order to give this power stroke, 1 ATP is hydrolyzed. 1 ATP is hydrolyzed. 1 ATP is hydrolyzed. If 1 ATP is hydrolyzed, it is broken down into ADP and AP phosphorus. Now, then the power stroke is given using 1 ATP. Okay, using 1 ATP. After that, after giving the power stroke, the Z line also moves. The Z line also moves from its initial position. Now, the head, after finishing its ATP, and now power stroke is completed. Power stroke, it has given power stroke. ATP is released. Now ATP is hydrolyzed. After completing the energy given by ATP, this head again gives a recovery stroke. Now here get the words. Power stroke. Power stroke. Pulling the actin towards itself, it gives a power stroke using 1 ATP. After ATP is completed, now no ATP is present. Now this gives a recovery stroke. It gives a recovery stroke. What is recovery stroke? The head again gets back into its original position. Its original position. Now how this happens? Here ATP is hydrolyzed. ATP is hydrolyzed. It forms ADP and 1 phosphorus. Now this ADP and phosphorus is combined in order to give a recovery stroke. In order to give a recovery stroke. Okay, understood? Understood? Yes, understood. Clear? Yes, it is clear. Is it correct? Yes, it is correct, absolutely. Now let's see the conclusions of this sliding filament theory or mechanism of muscle contraction. When the muscle contracts, what all decreases, what all remains in its original position, what all increases in length, everything let's see now. Yes. Conclusions, these are very important. Muscle. Yes, we are got to complete. No problem, please wait a little bit. If I tell you this conclusions, it is completed. In tomorrow's thing, we will be discussing about Cori cycle. We will be discussing about Cori cycle. So I'm really extremely sorry for that. There was some call. Yes, these after this in next tomorrow's session we will be discussing about Cori cycle. Yes, that Cori cycle is very simple. We will be completing the Cori cycle and then we will be looking at skeletal muscles. Yes, skeletal. Yes, more muscles completed. We will be going into bones. Okay, we will be going into bones. The first conclusion is length of sarcomere decreases. Length of sarcomere decreases. Z line contract, length decreases. Second one is length of H band is also decreased. Length of H band. What is H band? Only myosin. Actin, myosin. This is H band. This is H band. What is full length of myosin, which includes a part of actin also? A band. Okay, H band length also decreases, but length of A band remains same. This is very, very important conclusion. Remains same. Very, very important. Put 100 stars. A band remains same. Why? See, when the actin filament is sliding over the myosin filament, here A band includes the length of actin and also the length of myosin. Now if it slides over the thing, if it is decreasing its length, then actin also moves towards it. If actin is also moving, A band length remains same. Okay, A band length remains same. Repeated activation of this muscle. giving power stroke. Repeated contraction of muscle results in the release of lactic acid which accumulates and gives fatigue to the muscles. That is why these skeletal muscles are not fatigue resistant. Okay, hope you understood. So that is for this video. This is Vamsi signing off from BioLab. If you like the video, please hit that like button and also subscribe to our channel BioLab. And this is Vamsi. So in tomorrow's lecture, as I said, we will be talking about Cori's cycle, and also we have a part of muscles, types of muscle fibers. We'll be discussing that also. And yeah, I hope tomorrow I'll be again back with my tab. Okay, yes, that's it for this video. Yes, please hit that like button and also have a great day further.